A very important part of the cell cycle during interphase was S phase. And during S phase, we did have duplication of the DNA taking place. So another way to refer to this is that we had DNA replication occurring at that moment in time. And so if we talk more about that DNA replication process, the goal of DNA replication really was to take one copy of DNA or a certain amount of DNA and pretty much copy it so that we now have really twice that amount. So we go from having 1x, one copy of everything, to having ultimately 2 copies of everything. And more than that, we want these to be exact copies, because the goal here is that we want to end this whole cell cycle process with 2 cells that have exactly the same genetic content in them. So if we look at this process in more detail, we started with a double-stranded DNA molecule, and that DNA molecule was organized with a lot of different proteins inside the nucleus, and there's lots of different chromosomes there as well. So if we're just focusing on one of them, during this process, what is going to happen just overall wise is we're going to actually take this molecule and we're going to be breaking the hydrogen bonds that hold it together. So when we break those hydrogen bonds, what we will end up with is we will have the 2 strands that get separated from each other. And so if I just draw it like this, so that we can see how they've been separated from each other, the next step is going to be that we're actually going to build, and I'm going to draw it in green here, we're going to build the new copy of DNA right on top of those split strands. So the end result is that we end up with 2 double-stranded DNA molecules here, and these would be what we would refer to as sister chromatids. And these sister chromatids are meant to be identical to each other because we really took one double-stranded molecule, we split it in half, and we're going to copy each one of those and build the new copy right on top. So we have 2 of them, and when we talked about this inside the cell, that was those chromosomes that now look like little X's because you had the 2 chromatids held together. One important thing to note here is that each one of these chromatids that we have has 1 strand that's old, and that's represented as the blue here, and they have 1 strand that is new DNA, and that's represented by the green. Well, we have a name for this type of synthesis where we get 1 old strand and 1 new strand, and we refer to this as semiconservative. So we call DNA synthesis or DNA replication semiconservative replication of the DNA molecules. And so this semiconservative model of DNA replication, that is what we're going to look at in a lot more detail. Notice we are going to, again, take what we have, that 1 old strand, and open it up so that we can build right on top of it, or in other words, it can serve as a template for the new DNA strand. Now, when we talk about DNA sequence, we tend to just write the letters, and these letters would be the bases that make up the strand of DNA. So here I've just written some generic DNA sequence for us. We're just writing the bases here because the sugars and the phosphates for each one of the nucleotides that has these bases is going to be exactly the same as each other. And what I've written here is really just 1 DNA strand. But we know that DNA is always going to be a double helix. So even though I've written 1 strand, we know what the other strand is going to be. So I could go in here and I could write that other strand directly below it. And I am going to write that other strand anti-parallel. So I'm putting on my 3' and 5', and then I'm just going to write the correct base pairs across from the bases that I've already written up there. So what I'm doing is I'm putting an A across from every T, and I'm putting a C across from every G. So now we're looking at a DNA molecule in a lot more detail because we're seeing the actual bases that are present. And don't forget that these bases would be held together by hydrogen bonds. We would have 2 between all of the A's and T's and 3 between all of the G's and C's. So I'm just going to write those in for the moment just to remind us that they are present. Now, as we talked about, when we do have this semi-conservative DNA replication, the first step is going to be to separate. So we do want to separate the strands, and the separation of the strands would mean that we are breaking the hydrogen bonds. So if I draw the end result of that separation down here, now we can see those 2 strands that are separated from each other. And what is going to happen in the overall process of DNA replication is that we will have a collection of enzymes that are responsible for looking at each one of these old DNA template strands. So we are going to call these templates. These enzymes are going to look at them individually, and they will build new DNA directly on top. So if we just looked at the top DNA strand here, and we built directly on it, these enzymes will see the A and they will incorporate a T across from that. And they'll see a T, they'll put an A across from that, and so on. So I'm just going to go through and write this in for us so that we can see what we would end up with if we were just looking at this strand. We could do the same thing on the bottom strand, and again, if we put it anti-parallel, the enzymes, if they knew the base pairing rules, they could build directly on top of this. And if we did this separately, notice that the end result, although some is shown in blue and some is shown in black, if you look at this molecule and you compare it to this molecule right here, they are exact copies of each other. So that is really what we want to happen with this overall process. These are our 2 sister chromatids that are identical to each other in every way, because we took that initial template, we separated it, and we built the DNA right on top of it. Notice as well that each one of those double-stranded molecules has 1 strand of old and 1 strand of new.